Well, as Valentina introduced very kindly, the topic of my talk is genome editing. I know that maybe you are not so familiar with this topic, guys, so I will try to be very, very basic, but please interrupt me if something is not clear. I will be more than happy to discuss with you and to provide any clarification. Unfortunately, when I am in this full screen modality, I can see your raised hand, but Carmelo, Valentina, please, if you see something, feel free to interrupt me in any moment. So, because I know that we are not familiar with the topic, as I told before, let's start with some ontology. Okay, so let's start with some definition. So what you can see here is the canonical process for express one gene in one cell. Okay, so we start from the information which is in the genome, for example, the human genome. Okay, what you can see here is one sequence encoding for one specific protein. So this is the gene. Well, so what happens is that one enzyme named RNA polymerases starts the transcription process. This means that starting from the gene, we have the messenger RNA. Then the messenger RNA, just let me use the pointer so you can follow me much better. So the messenger RNA is then translated.
Vittoria, excuse me, I can barely hear you. I don't know if it's just a problem of mine.
Oh, I don't know. I will try to.
Okay, sorry, probably is
what
connection.
Now? What now?
Yeah.
Is a little bit better now?
Yeah. Thank you.
Okay. Okay. If something goes wrong, Valentina, let me know. I will try to change the connection. Okay. To connect from my mobile phone if something is not working. Okay. So what I'm saying that starting from the gene onto the genome, that this enzyme transcribed in the process of transcription the messenger RNA. Then the messenger RNA is translated by the ribosome into the protein. Okay, so in a situation in which we have a mutation at the level of the genome, okay, so we have a mutation in one gene, what happens that we have the expression of one protein which is dangerous for the organism. So what we want is to switch off the expression of this protein and to switch off the expression of this mutated protein, which is very dangerous for the organism because it causes some pathologies, disease, cancer, whatever it is. To switch off the expression of this protein, we have different options. Well, we can play at the level of the messenger RNA, okay? And when we do this, we talk about a gene knockdown, which means that we switch off the protein acting onto the messenger, but not onto the genome. The other option, of course, that we have is a switch off the gene, so to act directly onto the genome. This is a permanent modification, and we talk about a gene knockout, okay? When we talk about knock-in, what we mean is that we insert in the genome, for example, the human genome, a new gene, a coding for a new and exogenous protein. Okay, this is the knock-in. Well, if everything is clear, because in the following slides we'll talk about knock-in, knockout, knockdown. So if this is clear with you guys, we can start with the topic of this discussion, which is gene editing. To perform gene editing means to act at the level of the genome, so at this level. So this is the era of genome editing technologies, guys. The most popular one is the CRISPR-Cas9 system. What is that? It's a molecular scissor, which is literally able to cut the DNA. So this is how it works. Actually, we have one complex. The name of this complex is ribonucleoprotein. What this means? This means that we have two components. One is the RNA, the orange one, which we call the guide RNA. And then we have the protein, the Cas9, the green one. Okay? So the guide RNA, why guide RNA? Because this RNA is able to guide the enzyme into the locus, into the genome that we want to edit. How this is possible? Because this guide RNA is designed by the scientists in order to pair, you see a perfectly pair between bases, between the basis of the guide RNA and the genome, the target genome, the target locus. Okay, so what happens is that after this pairing between the RNA that we design and the gene that we want to cleave, after this perfect pairing, what happens is that we can activate the nuclease activity of the protein, meaning that the protein, like a scissor, is able to cut the DNA, to perform a double strand cleavage, okay, a double strand break. What happens after this break? You see, the enzyme is able to cut the DNA, but after this double strand break, what happens? Well, what happens is in the cells, we have some system that works as a self-repairing machine. And this self-repairing machine can, let's say, close this break. And there are two options for doing this. One is the system called non-homologous end joining. So this self-repairing system is not high fidelity. What this means? This means that when it repairs the DNA, it introduces some mistakes. What does this mean, mistakes? It could delete one base or could insert one base. So if you perform a deletion or an insertion, this is a mutation in the gene. And which is the consequence? The consequence is that we change the reading frame of the gene. So the consequence is that we switch off the expression of these genes because we express a trunked version of the protein or we don't express the protein at all. So we are switching off the expression of the gene. This is a gene knockout. Well, the other option that we have is to use this other machinery, which is the homology directed repairing machine, by using a template DNA, which can be the other allele, or it could be a gene exogenously inserted into the cells. By using this template, we can insert in this break the sequence encoding for one gene. Okay, this is gene knock-in. Okay, I want to stress, guys, one thing. By performing a deletion on a mutation, a deletion and insertion, what we obtain is a mutant organism. This is a mutation. This is a mutant organism. But if we insert an exogenous gene, we are creating an OGM, a transgenic organism. This is important because I know from a regulatory point of view, it makes difference to have a mutant or a transgenic organism. Okay. Well, now what I want to show you, a little cartoon, just to reassume a little bit. I want to be sure that you really understand how this system is working. Before watching this cartoon, I want to tell you that this system, the CRISPR-Cas9 system, this molecular scissor for cutting the DNA, is a big revolution in science. I should say, the bigger revolution in molecular biology in the last decade. And because of that, these two women scientists, the Emmanuelle Charpentier and the Jennifer Doudna, were prized by the Nobel Prize for Chemistry in 2020. Okay, let's give a look to this animation. Okay, here. Well, what this animation is showing you is a schematization of this enzyme. You recognize the two components. You remember, guys, I told you that you have the protein component, the Cas9, the blue one, and you have the guide RNA that works like a tom-tom, which directs the enzyme in the point in the human genome that you want to cleave. So you see the enzyme is scanning the DNA, literally scanning the DNA. At some point it stops. And what you can see here is that enzyme is unwinding the DNA double helix. Then one strand of the DNA is pairing to the guide RNA. And when the perfect pairing is happening, this activates the nuclease activity. You see, the enzyme cut the DNA. Then you have the self-repairing machine that try to repair this, let's say, this cut. But this create some mistakes. Then you see switch off the gene. So this gene didn't encode anymore for a protein or for a function protein. So we are switching off the expression of this gene. Okay. So if this is clear, now let's focus on the application. So what I want to show you today, guys, is which are the application of this technology, but also to discuss all together about the limitation of this technology. But let's start with the application, the potentiality of this, the potential of this technology. So how we can use this molecular scissor? For sure, we can use this molecular scissor for producing, for example, more plant variants. We can modify, we can create a new strand, for example, that resists to the climate change, or resist to the infection, or resist, I don't know, to the pollution. Okay, this is very important in agriculture field. Or we can create a genetic model. For example, if we have one human disease, which is caused by a specific mutation in one gene in the human genome, we can create with this system the same mutation in one animal model, for example, mouse model. Then this model recapitulates the pathology of the humans. And so we can test new medication, new drugs in the mouse model to see how the model reacts to this therapy. And if this is promising, then we can test it on humans, okay? The big challenge, let's say the dream, is to use this system for curing incurable human disease, okay? Then let's see which is the state of the art. Let's start with the application, for example, in the agriculture field. We have, let's say that the dream comes true, because if you see from the date, you can appreciate that in the end of the last year, the same crispant plant came into the market. This is a crispant tomato that has been launched onto the Japanese market. This is a crispant variant. How is, for which reason we performed the editing? The editing was performed to increase the level of this GABA, which is a gamma aminobutyric acid. Why we want to have more higher concentration of these nutrients? Because this helps to decrease the blood pressure. So it's a very important nutrient, and this is why the Japanese scientists produces this crispant tomato and they put on the market. What about the Europe? Let's say this is on the way. the most the country, let's say, in the most advanced stage is the UK. In UK, there are many trials that are testing CRISPR with, and I guess that in few months, maximum 1 year, we will have this on the market, on UK market as well. Okay. Well, but what about the therapeutic application? Also, the therapeutic application of this new technology are practical application. But I should say that the number of application, the potential number of application is uncountless. But the real number of real application is very limited, because there are some limitations in this technology. There are some issues that we want to solve, and this is, I want to focus on that. But before to start with the practical discussion, let's make an introduction. How we can use the CRISPR-Cas9 system for make therapy on humans. Actually, there are two strategies. One is the in vivo strategies. The second one is the ex vivo strategies. So what means ex vivo? Ex vivo means that we can harvest some cells, specific cells, for example, blood cells from the patients, or immune system cells from the patients. So we harvest these cells from the patients. Then we insert the machinery for the CRISPR-Cas9, for the genome editing into these cells. Then we perform the gene editing, and after the editing, we can reimplant these cells into the human body. So this is ex vivo, because the cells are taken from the patients, modified, then reimplanted into the patients. This is considered a safer approach, because not all the cells are edited or are exposed to the editing machinery, but just those that are harvested. And so we didn't expect any side effect on any other cells in the human body. Conversely, in the in vivo strategies, the system for the genome editing is injected systemically. This means that in theory, all the cells on the human body could be exposed to the treatment. And this raises some ethical concern, because I will tell you in a while which are the limitations. Well, actually, there are two limitations. One is safety, the second one is delivery. Okay. So let's explain why we have safety issue with this technology. Again, I have a short video that we can comment together. Okay. So what you can see here in this video is the enzyme, the CRISPR-Cas9 enzyme, and the DNA double helix. As you remember, what happens is that the enzyme undouble the double helix, pair with the target, then cleave the target. And this is a perfect pairing. What happens is that sometimes the guide can bind to sequences that are similar but not identical to the target. And in any case, this can activate the cleavage. I will go back for a while, for just a few seconds, to show you this again. Okay, you see here that the guide RNA is pairing with a sequence that changes just for one base from the target. So it's similar but not identical to the target. So the guide could bind to this sequence that is in another point in the human genome, which is similar but not identical, then can cleave this sequence. So this is an off-target event. What this means? This means that this molecular scissor is cutting one sequence, another sequence in the human genome, which is similar but not identical to the target. Okay, because it changed, for example, for 1 base pair, 2 base pair. Okay, this is very toxic, very dangerous for the organism because this is an unwanted cut. You can cut one gene that is essential for the survival of the cells, or in the worst inconvenience, you can also cut a gene, and cutting these genes, this is responsible for oncogenesis. Okay, so you can create a disease rather than curing a disease. So this is a big problem, this off-target event. I should say that the probability to have the off-target event is very, very low if you compare this to the probability to have on-target event. But even if low, it's not a null possibility. Okay, at some frequency this could happen, and if it happens, it's very, very dangerous for the patients receiving this treatment. So this is safety. The second problem.
Excuse me,
yes,
Vittoria.
sì.
Excuse me. When you talk of low probability, how can you precisely quantify?
Yeah,
what do you mean by low?
That's a change by gene and sequence by sequence. But just to give you some numbers, I could say that if you inject, okay, one machinery in one cell, you could have the 80% of probability to cut the target, the on-target sequence. And you could have the 0.5% of probability to cut an unwanted target. Okay. Which means that if you have 200 cells, you can cut at least one cell in an unwanted point.
Exactly.
And this could be just what that single cell, if cutted in unlucky events, could be the beginning for oncogenesis, because it could replicate in a crazy way and start a mass. Okay. It's just one cell among 1,000, but depend on the place, this could be dangerous for the organism. It's a very, let's say, low probability events, but you know,
well, not so low actually.
Not so low, but especially depends the patients that you have. If you have one patient that has a so severe disease that is going to die in 1 year, you accept this risk. In other specific case, you couldn't accept this risk. Okay. That, you know, that's ethical problem. You know.
Thank you.
At the end of the story is always ethical problem. Well, thank you for the question. If there is any other question, guys, please interrupt me. Well, the second point is the delivery. Because this delivery issue is also related to the previous, the safety. Because in principle, if we can target just the disease and not to target the healthy cells in the human body, we can also find a solution to the safety issue. Okay, so this is the big problem. Try to deliver our medication just to the diseased tissue and not to touch the healthy cells. This is not easy. This is very challenging. What the scientists do, according to the best of our knowledge, the best of our technologies, is to use viral vectors. Because inside the genome, this virus could be modified to insert all the genes that encode for the CRISPR-Cas9 machinery. So they are the carriers, so this viral vector are modified to express the machinery for the genome editing. But using viral vector is not so safe, have some safety concern. Okay, you know, these vectors are used also to perform gene therapy. and there was problem in the past with the gene therapy. So this is why scientists are trying to find solution, alternative solution. This is again one cartoon that explains why this is so challenging. So what if we consider here is to treat a disease at the level of the retina. For example, retinal cells. We have one gene mutated, and these genes, this mutation causes a pathology in the retina, in the retinal cells, for example, photoreceptor. Let's try to figure out which is the travel that the medication should do for reaching this photoreceptor, okay? So we can give this medication as an eye drop. Then these dry hops should reach the posterior chamber where the retina is located. Then should target the photoreceptor. Then should cross the cell membrane, the nuclear membrane, reach the DNA, find the gene into the DNA. So it's a very, very long trip. So many times the drug fail, not because it's not able to cleave the DNA, but it's not able to reach the DNA. That it's a different problem, and this is the delivery issue, okay? So what scientists are trying to do is to solve this issue, safety from one side and delivery from the other side. And this is the reason, because these issues are still open, this is why potentially we could apply this technology for treating 1 million of diseases. This is in principle, in theory. But because of this issue, if you give a look to the table of the clinical trials that are testing this CRISPR-Cas9 system, this table is very short. Okay, so there is this is the problem. In theory, we can use genome editing for cure any incurable disease. Practically, we have still problem related to safety delivery, and this is why, yes, the CRISPR-Cas9 system is entering the clinical trials, which means the trials on humans, but the list of trials is very, very short. Now, what I want to do to show you guys is to show you which are the major clinical trials, how they use it, which pathology we want to target. I don't want, if you want to have just 5 minutes break now, I can continue and stop later. What do you think, Carmelo?
Actually, for me, it's okay if you can continue. Valentina cannot hear you. Valentina, your mic is mute.
I suggest to go on if Vittoria doesn't mind, of course.
Of course, we can go on. So let's continue and let's give a look to the different clinical trials that are ongoing. This is one. Okay, the name of this trial is CLIMB. This trial would provide a new solution for the treatment of severe sickle cell disease. As you know, severe sickle cell disease, anemia falciforme in Italiano, is a very severe pathology because the red blood cells couldn't express hemoglobin. They couldn't fix oxygen, and because of that, these poor patients are dependent on continuous transfusion. So it's a very severe pathology. How the CRISPR-Cas9 system could help? What the scientists the science knows in the field is that the humans could express two forms of hemoglobin. One is a fetal form, and one is the adult form. So we possess just the adult form because the fetal form is not anymore expressed after the birth. Okay, why? Because there is one gene which acts as a repressor and switch off the expression of the fetal form. Okay, so you understand which is the brilliant idea. The idea is let's repress the repressor, because if you repress the repressor, then you can activate the fetal form. Okay, And if we activate this fetal form, these poor patients could have their own fetal hemoglobin. So they didn't depend anymore on the transfusion. So this is exactly what the scientists did with this system. They used the CRISPR-Cas9 system to switch off the repressor. So this is the workflow of the trial. So this is the patients. We can collect the precursor cells, those cells that after differentiation gives red blood cells. So they collect the precursor cells. Then they perform the genome editing in order to switch off the expression of, in order to switch off the expression of these genes. After doing that, they reimplant the cells. Then these cells can differentiate in the red blood cells, and these red blood cells can express this fetal hemoglobin. So the patient has his own fetal hemoglobin. It would not depend anymore on the transfusion. So this is a cure for incurable disease. You understand how much is important. I want to stress, I want to highlight that this therapeutic approach is ex vivo. You know, it's not really in vivo, it's ex vivo. So we harvest the cells, treat the cells, then reimplant the cells. Another ex vivo approach is this other clinical trial. The name is Carbon clinical trial. In this specific trial, our target are T cells. T cells are immune system cells, and the pathology that we want to target, we want to care, is the cancer. So you understand how much is important, which is the idea. These T cells that I told you are immune system cells are edited in order to express one receptor, the CAR receptor. This is a knock-in because we are inserting a new gene. And this gene expressed for this receptor. Why is this important, this receptor? Because this receptor is able to bind a ligand which is expressed by the cancer cells. So by doing this, by implanting these cells into the patients, these cells can, let's say, recognize the cancer cells, bind the cancer cells, and then kill the cancer cells. So this is a cancer treatment called immunotherapy. Okay. Another example.
It's a bit. It works a bit like a vaccine, doesn't it?
Yeah, it's something we fight the cancers from the inside. We use the same cells that they kill the pathogens. We use those cells to kill our cancer cells as they are pathogens. So it's a very smart idea. Well, this is another treatment. The name is EDIT 101. This is for the treatment of the Leber's congenital amaurosis type 10 disease. This is a disease at the level of the retina that causes blindness. The patients that suffer from this pathology, so these poor patients has a mutation, this gene, centrosomal protein 219. And because this protein is mutated, they cause degeneration in the retina, and so the patients don't have any more the vision. So the idea is very simple. Let's switch off the expression of these genes. Let's perform a knockout of the gene. So if we knock out these genes, then the retinal cells can regenerate, partially regenerate, and so we have a recovery of the vision. This is what has been demonstrated by these trials, that the patients could recover the vision. I'm mentioning this trial because it is interesting. In this specific case, we have a local treatment because this drug is injected in the eye, is a subretinal injection. So you see, this is something in the middle between ex vivo and in vivo. It's not really in vivo because you don't inject in the bloodstream, but it's never ex vivo because you don't transplant cells. But it's a local injection This is still considered safe because the drug remained localized into the eyes, so didn't touch other cells in the human body. There is, at the best of my knowledge, just one trial that tests the in vivo paradigm. So this trial is this one, and this comes thanks to advance in the field of nanotechnology, thanks to nanomedicine approach. I will explain you in a while. So which is the pathology? The pathology is another very severe pathology, is the transtitherine amyloidosis. Transtitherine is a protein which is secreted by the liver. And this protein is able to execute its function when it is in this form, which is a tetrameric form. Okay, you see 4 subunits that assemble all together to form this tetrameric form. In these patients, this protein is mutated. So the protein is not able to tetramerize, but it, let's say, precipitate in the form of amyloid fibrils. And these fibrils can precipitate in the liver, but also in the other tissue, especially in the heart. So it's very toxic for the cardiac muscle. And because of that, the expectancy of life for these poor patients is just 5 years of life. Okay? But the scientists produced a nano formulation of the CRISPR-Cas9 system, which is able to target this gene. This is a lipid, you see, this is a lipid nanoparticles that contain the machinery for the genome editing. This is injected in the bloodstream. When it's injected, what happens is that this lipid nanoparticles suddenly is coated from specific ligand, ApoE protein. These ligands are able to bind to the hepatocyte. And so all these particles are accumulated in the hepatocytes. There, they can edit the DNA of these hepatocytes, and thus this is very the therapeutic efficiency of this medication is very, very high. Actually, one single injection is enough to cure these poor patients. Okay, so it's very, very efficient, and it's also safe thanks to the nanomedicine, because we can inject this formulation into the bloodstream, but because of the nanoparticles, they accumulate just in the hepatocyte. This means that any other cells in the human body is a very low exposure. It's not null, but is a low exposure to the medication because all the drugs accumulate in the liver. Well, as I told you before, the potentiality, the potential of this technology is huge and countless. Any uncured disease could have a therapeutic approach with the CRISPR-Cas9 system. But as I told you, the list of ongoing clinical trials is short because still we have problems related to safety and delivery. And the scientists are trying to solve this problem. Of course, the science never stops. We are trying to move forward to find new solutions to solve this issue. Here you have a short list of the different research lines. There are people selecting high fidelity variants, people designing non-viral vectors, switchable system to perform knockdown rather than knockout, and base editor. I want to discuss very shortly about these options. Let's start with the high fidelity variants. What means high fidelity? High fidelity means that we want to select a variant of the Cas9 protein that have higher fidelity. That means are just able to bind the target, so perfect pairing, but they didn't bind when you have not such perfect pairing. In other words, you want to select a variant that just let happen on target event, but not off target. There is one Italian group, the group of Professoressa Cereseto, which is very active in the field, and she was able to demonstrate that one variant, the name of this variant is EVO-Cas9, is able to work in this high fidelity modality. What you can see in this graph is that the normal, let's say the normal wild type Cas9, for example, when injecting in one cells and targeting one specific genes, was responsible to perform of 28 off-target events. By using these variants, the number of targets are not 28, but decrease at 12. So what scientists are trying to do is to improve this in order to reach 0 off target. So this is, let's say, the big challenge of this research line. Another option is to work onto the delivery. As I told you before, the only chance that we have to apply for the in vivo approach is to use nanomedicine. Why? Because nanoparticles have this are the magic bullet that can target medication to specific cells. So what I showed you before was the example of lipid nanoparticles, but there are many scientists that are working also with different particles, for example, polymer nanoparticles or DNA cages, which means particles made by DNA. There are other scientists proposing exosome. Exosome are vesicles produced by cells to communicate with other cells. So what these scientists proposed is to use this as system to deliver the CRISPR-Cas9 system. And there are other scientists working on gold nanoparticles. My team is working on that as well. So I will give you a practical example on that. Okay. So let's discuss about this example. I'm talking about a switchable system. What means a switchable system? It means that it is a medication that could be is inactive and could be activated. Which is the big advantage of this approach? The big advantage, guys, is that you can give to the patients the medication in one form which is not active. Then you can activate the drugs just in the tissue, in the diseased tissue. And the big advantage is that all the other cells in the human body receive the medication in one form which is not active. So there is no side effect. Here what you can see is a cartoon. So what you can see here is the bloodstream, and there are these nanoparticles that are flowing in the bloodstream. These are not active, but they are photoactivable, which means that if you expose this particle to a laser radiation, you can activate them. So what you can see here now is a patient. So this patient is a cancer, a melanoma cancer. So there is a lesion in the skin that we want to remove. So we can inject these particles in this form, which is not active. Then we can expose the lesion to the light. Just in that point, only at that specific point, we activate the formulation and the genome editing. This means that we can kill the cancer, but any other cells in the human body is receiving the medication in one form which is not active, so not dangerous. Okay. This is exactly what my team is doing in this research project that Valentina mentioned in the introduction. So we have a project ongoing that has this specific aim to provide this light switchable nanoformulation. So this is a nanoformulation based on gold nanoparticle, which can be activated by light. So why nanoparticles? From one side, we can use the nanoparticles to target the medication, to cross the cell membrane, to cross the nuclear membrane, and to reach the nuclei. On the other side, we can activate these particles with the light, and so we can make the treatment switchable. So the name of this project is iGene, has been funded by the European Commission, Horizon 2020, under the Future Emerging Technology scheme. I am the coordinator of this project, and there are many other partners. Many of them belongs to the industry because they are trying to provide a new solution, technical solution, and put on the market this technical solution. Okay. Well, I don't want to tell you more about that because it becomes too much complicated, in my opinion. I want just to conclude with the list that I give you before, just to mention this alternative approach that is going on in the literature, which is the knockdown. Remember what I told you before. The idea is to replace the knockout with the knockdown. Just a little reminder. You remember what we told at the very beginning of the lecture? The DNA is transcribed in the messenger. Just a second. The DNA is transcribed in the messenger mRNA, which is translated to the protein. As I told you before, if we kill the gene, we perform the knockout. But if we kill the mRNA, we perform the knockdown, which is safer because this is not a permanent modification. We didn't touch the DNA, but just the product of the expression of those DNA. And so this is not permanent, so it's safer. How we can do this? We can do this in two ways. One way is to use the interference. For doing interference, the scientists have developed one variant of the Cas9 system, which is the dCas9 system, which means that this enzyme is still able to unwind the DNA, to pair to the target, but it's not able to cut. So the point is how we can make use of that if it's not able to cut. The idea is that, you remember what I told you, that the expression of the messenger RNA happens because there is an enzyme, which is the RNA polymerases, which bind to the gene and transcribe the RNA. So the idea is to target this enzyme to the gene in order to avoid the RNA polymerase to bind to the gene by steric hindrance. Okay? In other words, if you put the enzyme onto the DNA, the polymerases couldn't bind, and so couldn't express the mRNA, which means that you are not expressing the protein. Okay? So this is a brilliant idea. Another idea is to create some variant of the Cas9 and exists, the name is Cas13. This Cas13, while the Cas9 is able to cleave the DNA, the Cas13 is able to cleave the RNA. So when you inject this system into the cells, it can bind to the RNA and cut the RNA rather than DNA. And there are also practical applications of this technology that is became more and more popular. The most popular application to use this technology to cut the mRNA of coronavirus, COVID-19. And this has been proven and published by the literature. Okay. Final, the last discovery in the field that was really an amazing discovery is new enzyme that rather cutting the DNA, they can replace the base. So they can replace one cytidine with a thymine, and they can replace one adenosine with one guanosine. So they are replacing the base. Why this is important? You know, many pathology are caused by point mutation in the human genome. If you can, like if you have a red pencil, like a teacher with a red pencil, if you can delete a mutation, change the base, replace the base with another one, you are removing the mutation, so you are removing the disease. So this is very at the beginning, this research is very at the beginning, but they expect that in the next 5 years, this will represent a big revolution in the field. So this is why I want to mention you. So time for the conclusion. We have the technology for knocking out a gene, knocking in a gene, knock down a gene, edit a single nucleotide, which means replace one base with another base. We can create a new plant variant, but of course, if we create a new plant variant, we should also consider issues, such as what about allergy, biodiversity? We should consider this issue. We can cure incurable disease. We can edit somatic and germinal cells. We have this technology. So I want to leave you with this question. We are really ready for this, or it's really a lot what we can do? And we still have to understand how to handle this. So this was really the conclusion. I hope I stay in time. I hope that this was not too much for you guys. And
Thank you, Vittoria. I've heard from you quite a few times, but ever again I am humbled, let me say, by the beauty and importance of this research. The question, Vittoria, with Vittoria left us is a typical legal and ethical question. So I wonder whether some of you, are we ready for this technology? There are more than a few ethical and legal issues connected to, I don't want to say the correct answer to this question, but an appropriate answer to this question. And I'd like to hear it from you, based on your knowledge, on the best of your knowledge. If you have doubts or curiosities and so on, based on your legal knowledge, of course.
What do you think, guys? Are we ready for that?
Yeah.
Any comment on that? From any point you want to discuss this, ethical, regulatory, social point of view. Of course, you are students, but you're citizen.
Exactly.
And you should also consider this from your point of view. You could have friends or, I don't know, yes, friends, maybe they could need this such therapy. Which is your opinion on that? Don't be shy, guys. Try to make any comment on that. That's difficult to have an opinion, because of course any opinion is right in this context.
Yeah. I think that there is no correct answer, but it is appropriate to think of this, because it is really a revolution. I was hearing a few days ago a beautiful series of a podcast on the BBC on artificial intelligence. You can find it. The read lectures, and the speaker was Stuart Russell, who teaches at Berkeley and is one of the most eminent experts of artificial intelligence, and he mentioned these technologies together with progress with artificial intelligence systems as the defining feature of 21st century societies. It is not far from the world we live in. It is actually the world we are living in now.
It is. Absolutely, it is. It's a reality.
And for instance, there are a number of problems concerning constitutional admissibility of at least certain application of CRISPR-Cas9. There are also problems related to the
I see the comment
risk
of
management and
yeah,
so on.
Of course. Michaela is writing, from a legal point of view, there are certainly some liability issues, of course there are. But what I want to say, you guys, that I have many opinions, but the first one is that science never stops. I mean, my point is that all the scientists working in the regulatory, ethical, legal framework should help the scientists to manage this point. But science should not stop. Of course, we need a framework that support and make a better use of this technology rather than stop it.
Yeah, exactly. I don't know if it is appropriate in your opinion, Vittoria, but sometimes I'm thinking of it as an equivalent to atomic energy. You know, we managed with a huge effort to control the military use of atomic energy, and without at the same time removing atomic energy research. And I don't know if it is appropriate, this analogy for you.
Yes, in my opinion, absolutely it is. You see that the story sometimes repeats itself. Yeah. Of course, there is this problem with the nuclear energy, from the past, what it could happen if you don't have controls on the use. But on the other side, if you speak about nuclear energy, you should also talk, for example, about radiotherapy. No, if we don't have the nuclear energy, we don't have a radiotherapy. And how millions of patients can take advantage from the use of radiotherapy for killing cancers. So can you really say that this is not good for the society? So it's exactly the same point. The problem is not the technology, it's the use and how to limit the use of this technology. So I think that the consideration that we can apply to this example also apply to the gene editing. Of course, if you don't put limitation, you can have many consequences. People can use this for military use, for man enhancement, or for, many unwanted use, of course. So it's very, very dangerous. But the problem should be solved at the political level. We need some some guidelines that should be not be at the European level, but worldwide level. And communicate worldwide is not easy to have common strategies worldwide for handling such issues.
It is important to also to consider that there are actually standards. There is a movement for standardization of genome editing technology. It is in progress. So we have an ISO standard, an ISO standard for. We have a number of genome editing standards yet in force. Carmelo.
I have a question from, let's say, technical perspective that probably have some legal also application, a legal impact. So regarding the tomatoes, so my question was, are these tomatoes a new species? So you have like seeds of these tomatoes able to have a higher quantity of GABA, or each plant was modified later? So from technical point of view, it is a new species or like something that you modified after?
It's a Sicilian variant that is permanently modified to overexpress GABA, because GABA is already expressed by tomatoes. It's an overexpression of GABA. But it's a variant that you can culture in the field. And so this is why I'm talking about biodiversity. Because if you change the equilibrium, at some point you could have some species that replace other species because maybe they are more resistant strain or whatever. So there is an impact, an environmental impact on that. But Japan could allow this because this is considered not OGM, it's considered a mutant strain.
Okay.
So you see that there are countries that are working on that, but this is a global problem because it doesn't make sense if Japan could culture these tomatoes and any other part of the world couldn't, because the effect from Japan spread worldwide. So this is why I'm telling that the decision should be at the world level. Otherwise, it's a nonsense.
Okay. Thank you.
I don't know if this was answering to your question.
You say mutant and variant were, I mean, let's say enough for me to understand that you can have an impact, a non-local impact when you.
Exactly.
So
just a final point. Of course, as Michaela suggested, there can be liability problems. There are always liability problems connected to new technologies. But I suggest you to reflect also beyond liability, because liability, of course, is a welcomed and perspective, but it is by no means the only relevant perspective. Liability suggests that the problem is one-to-one. The victim of a erroneous application of the technology and the team of physicians or applying it. And the problem is bigger than this. It involves society in general. So liability is one of the issues we must consider but by no means the only issue.
Yes, absolutely. This is
and
the point.
we will say something more tomorrow, because this is a specific. Tomorrow I will talk about informed consent, and yes, this is informed consent at the preclinical stage or clinical stage. Is informed. We are not performing trials, medical trials for in vivo applications, but we will probably in a few years, I think, and so there are specific problems with this.
Valentina, there is a beautiful comment from Serena Fedi. I don't know if you want to read to it.
Yes, please. All the unknown is frightening, but with knowledge, you may be able to calm the distrust of technologies that are so revolutionary. Of course, this would improve the quality of life of people who, unfortunately, develop cancer diseases even at a young age. My little thought, however, is that if, from a genetic point of view, this activity must undoubtedly be controlled to avoid manipulations of another type, which from a noble cause ends up in another. Exactly. This is exactly the point. So, with this comment, if there are no questions, these beautiful comments by Serena. Oh no, there is another comment. In China, everything is possible. They allow cloning. They build COVID hospital in 1 month, etc. Yeah, well, it is not exactly that they allow cloning, but yeah, we will talk about the He Jiankui experiment tomorrow, for instance, and yeah. Still, I think that China is not the place we want to end up at the end of the day. Like, the Chinese approach to revolutionary biotechnology is a bit, how can I say, disinhibited, so to say. Democratic societies need more control.
Agree.
Is there any other? I think it. Why don't you speak out instead of writing messages? Michaela again says, I think it can help many, but it can also hurt if it gets into the wrong hands. Therefore, there should be clear rules. Yeah, the question is, why? What do we mean by wrong hands? My idea is that it is wrong to think of some crazy scientist. This is not the point. I don't know. I think, to summarize my opinion, I don't know if Vittoria agrees. The point is not or are not exclusively the individuals, the single people. Of course, there are bad scientists and good scientists, but the point is institutions are more relevant.
Yeah, I would also add Valentina, society, because I think we tell many times we should decide which future we want for us, our children, and so it's
what kind of society we want to live. So if we decide, for instance, consider the definition of disease. At this very stage of the development of this technique, there is a huge consensus that CRISPR-Cas9 and another thing of CRISPR techniques should be applied only for therapeutic purposes. But still, there are a number of people, and believe me, they are not bad scientists or crazy guys and so on, that submit who submit that, well, why should not we use CRISPR in order to improve people, in order to get more intelligent, more physically brave,
or to extend the life expectancy.
Extend life expectancies beyond 1 century and perhaps more, and so on. And these applications have obvious limits of compatibility in a overcrowded world. People who live beyond up to 200 years, well, it is a problem. It has sustainability problems. Yeah, and there are also problems of democratic structure of societies. If we manage to develop a class of super intelligent people, well, this class for sure will want to be in control, and they will manage actually to be in control, and so on. And so we don't live in perfectly egalitarian societies, and I don't think it is even too hospitable to live in a perfect egalitarian society. But maybe we don't want to live in a society where the brightest and the best are determined before they are born.
Yeah,
I don't know. There are a number of problems with CRISPR-9 applications.
Yeah, the letter is speaking about discrimination. Yes, it's a form of discrimination.
Yeah. And also there is the problem, but this is I think this is a more general problem. We can observe it every day with vaccines. There is the problem of access to mature technologies. The Western countries will develop cures based on CRISPR-Cas9 before then.
Yeah, that's a very expensive. It's a very expensive technology.
They are very, very expensive, and so there is. But this is a problem that is not limited to this particular technology, actually.
Yeah.
But yes, there is a number of very, very critical considerations. Okay, if there are no other inputs from you, anyway, too much democracy in the West started to hurt development. Yeah, you're probably thinking of Novak's movements, alias, I don't know. But we can talk about this tomorrow. And thank you, everybody, and thank you, Vittoria, above all.
You're welcome. Big pleasure to stay with you.
Thank you.
All
you,
of
Vittoria.
you. Ciao.